All right, so I had mentioned in class some specific tools that you can use to study better. And I had mentioned here that you can prompt AI to make podcasts of your slides. So here I am going to show you how you're going to do that. So what you're going to do here, you go to your dashboard and you can click here on academic success. Actually, here you're going to find that supplemental instruction information, and I was looking to see if it is available for our class, and unfortunately this term it's not available. They're not having SI for this class. But if you're taking some other classes that you can find it helpful, you can go on this site here and say, okay, search availability for your course. So look here. Here they have Anatomy and Physiology 1. Okay, so I do have for this other class. Here is my session. So they do have availability. And look at that. You have also general chemistry and organic chemistry, biochemistry, and genetics, but they do not have our class here. They do not have cell bio this time. Oh well. All right, so now let's see here if we find the AI tools. Where is it that we have to go to find it? Let me go back to the dashboard. I know I found it here somewhere. Okay, so this page was not in my dashboard, or at least I could not find it there. But I'm going to put the link here. Okay, so you follow that link, fau.edu/AI. Okay, and you're going to come to this page. Now here you just scroll down and you're gonna do here AI tools. You're gonna click on learn more, and here you're gonna find the endorsed tools. I still don't know how to use this Copilot or Owlon, any of this, so I went straight to this guy here, Notebook LM. Okay, you get in here and you say access Notebook LM. So it's going to open this site. You're going to try Notebook LM. And here it's going to show me what are the notebooks that I have created. Okay, so here is showing the notebook that I started to create in the class. So remember, I had uploaded the PDF file. Let me see if I can look at the file here. Oh, they do their own AI thing for this file, so it's not that PDF file anymore. But I did upload that PDF file here. Now, if I open that file to show you exactly what I had uploaded, so here it is. Okay, I have here the total of 11 slides. And this is in the topic of model organisms. So I just stuck to this topic for these 11 slides. From here, I made a PDF file, okay? And I uploaded the PDF file to this notebook. After that, this AI will give me a brief summary of what the PDF file is about, and it's going to show me these 4 options. Okay, so I had this one before, which is the audio overview. So this is going to give you the podcast, which is right here. And let me see what happens here. You can do mind map, and it's going to generate, I haven't done that before, but it's going to generate whatever they call mind map. So let's see what happens here, what is doing its thing. I'm going to show you what happens with the podcast here, okay? Oh, look at that. It already did the mind map. All right, so this is Model Organisms. It's going to give you a definition. This is cool. Gene function and mutation. All right, that's really neat. Mutations reveal gene function, alteration in organism shape, and then the example. And here's going to tell you about the eukaryote, unicellular eukaryote, bacterium and viruses, key discoveries from E. coli, and then it's going to show you here animal cells, and oh, look at that, it has invertebrate models. It talks more about the C. elegans. Ooh, this is really neat. And then it goes for vertebrate models, and he's going to tell you what is important about each one of those. I really like that. Look at that. Oh, it's first time I'm using this AI. What are we learning? Look at that. This is really neat. I like this mind map. And you can zoom out. Ooh, neat. This is a new toy. Look at that. You zoom out and you see the entire mind map of this section of the class. Cool. So I minimize that. And now let's go back here to the podcast. Now you're gonna like this. Let's check this out.
Imagine trying to understand a massive, intricate machine by studying every single screw at once. Just overwhelming, right?
Completely.
But what if you could pick out a smaller, simpler version of that machine, learn its fundamental workings, and then apply those lessons to the big one?
Mm-hmm.
Well, that's pretty much exactly what scientists do to unlock the secrets of life.
It's a really powerful strategy.
You got these two virtual people who are talking to each other. You get the podcast here, which is about 20 minutes, okay? And you can listen to the podcast and get a better understanding of that section of the class. And I will put this podcast, this entire podcast, in this video, so you can listen to that, or you can create your own. But well, let's finish this podcast, and then you tell me what you think about it.
Imagine trying to understand a massive, intricate machine by studying every single screw at once. Just overwhelming, right?
Completely.
But what if you could pick out a smaller, simpler version of that machine, learn its fundamental workings, and then apply those lessons to the big one?
Mm.
Well, that's pretty much exactly what scientists do to unlock the secrets of life.
It's a really powerful strategy.
Today, we're taking a deep dive into the world of model organisms, these living beings that have been, well, instrumental in nearly every major biological breakthrough.
Absolutely fundamental.
From tiny bacteria all the way up to the humble mouse, these are really the unsung heroes of scientific discovery. So what are we diving into today? We're going to break down what truly makes an organism a model for science, understand why they're so incredibly vital,
Yeah.
and discover some of the most mind-blowing biological breakthroughs we owe to them. Get ready for some serious aha moments.
Looking forward to it.
You know, when we talk about life, we often think of humans or maybe complex ecosystems.
Sure, the big picture stuff.
Right. But the real pioneers in figuring out life's basic rules are often far smaller and sometimes, yeah, a little squirmy.
And what's fascinating here is how scientists have so strategically chosen these organisms. It's not random. No, they pick them specifically to isolate and study particular biological processes. It's this deliberate choice that lets us unravel the fundamental principles governing all life.
Including our own complex biology.
Exactly.
Including our own.
That makes perfect sense. So to kick us off, what exactly defines a model organism? We know it's a whole living being, whether unicellular or multicellular, but it's clearly more than just, being alive, right?
Oh, much more. There are specific criteria.
Okay.
It's far more than just being alive. These organisms are chosen because they possess a kind of combination of characteristics that make them exceptionally amenable to scientific study.
Like a toolkit.
Think of it like a biological toolkit. Yeah. They often have relatively simple body plans, incredibly fast life cycles. They're easy and, importantly, inexpensive to maintain in a lab.
Right. Practicality. Exactly. And crucially, they share fundamental biological processes and genes with other, more complex life forms.
That last point seems key.
It is. That's why studying a worm, for example, can actually tell us something important about a human.
That brings to mind something quite striking from the material, how mutations reveal the functions of genes. The example of the yeast, Schizosaccharomyces pombe, it's traumatic. A single gene change turns it from its normal cigar shape to a T shape.
It's a fantastic visual, isn't it?
It really is. But it makes me wonder, how exactly does simply altering one gene let scientists figure out its specific function so clearly?
Well, that image really highlights the elegance of the approach. By introducing a mutation, essentially, you're changing one specific gene and then meticulously observing the resulting alteration in the organism's form or function.
Okay.
Scientists can directly deduce what that gene does. It's sort of like removing one tiny gear from a clock and seeing which hand stops moving.
Ah, I see. Cause and effect.
Precisely. And this fundamental method, this logic, it underpins so much of genetics. It lets us unravel the complex instructions encoded in DNA piece by piece.
Okay, let's start really small then, right at the foundation of molecular biology itself. It's incredible to think that molecular biology began with a spotlight on one bacterium and its viruses.
Absolutely. The bacterium E. coli. It became the ideal model organism.
Why E. coli specifically?
Well, not just because it's simple, fast-growing, and easy to culture, though that helps,
Sure.
but also because it's so readily genetically manipulated. And its viruses, called bacteriophage or just phages, they were equally critical.
So the bacteria and their viruses together.
Exactly. Together, they form this remarkably controllable and accessible system for those early, really foundational genetic experiments.
I was absolutely fascinated to see the list of key discoveries from E. coli and phages. I mean, this stuff really laid the groundwork for so much of biology today.
It really did.
Take the famous Hershey-Chase experiment in 1952. Before this, lots of scientists still thought proteins carried genetic info, right?
That was the prevailing theory for quite some time, yes.
But Hershey and Chase, using these bacteria and phages, they provided the definitive proof. Elegant proof, actually.
Very elegant.
That it was DNA. And that just forever changed our understanding of heredity. I mean, wow. That was a massive paradigm shift.
A truly monumental finding, no exaggeration. And then you had Jacob and Monod discovering the lac operon.
The lac operon. Okay, refresh me on that.
So for those unfamiliar, an operon is like a genetic on-off switch. It lets bacteria regulate which genes are expressed or activated based on what's happening around them, like if a certain food source, lactose in this case, is present.
So it controls gene activity.
Exactly. This discovery explained gene regulation, this incredible process where cells decide which genes to turn on or turn off. It allows them to adapt, to specialize. It was the first time we really understood how genes are controlled.
And it didn't stop there, did it?
Yeah.
E. coli studies were also central to figuring out the genetic code, deciphering it, how those combinations of 3 DNA bases, the codons, specify which amino acids to use to build proteins.
Critical piece of the puzzle.
And let's not forget, molecular cloning and recombinant DNA developed using E. coli plasmids.
Those little circles of DNA.
Right, that bacteria can share. And phage vectors virus is basically repurposed as delivery trucks for genes.
It's amazing, really. And if we connect all these discoveries, these profound findings to the bigger picture, they aren't just historical footnotes. They are literally the bedrock of modern biotechnology, genetic engineering, our entire understanding of how genes work at the most fundamental level.
Wow.
They gave us the concepts and the essential tools to even begin thinking about manipulating DNA and designing new biological systems.
So E. coli gave us the fundamental building blocks for prokaryotes, those cells without a nucleus. But what about eukaryotes? You know, the cells that make up plants, animals, us, with their much more complex internal bits. How do we scale up?
That's where unicellular yeast comes in. Specifically, Schizosaccharomyces pombe is a great example. It serves as a minimal model eukaryote.
Minimal model, okay.
So while E. coli illuminated prokaryotic life, yeast provided this remarkably straightforward system to study eukaryotic cells.
Simpler than our cells, but still eukaryotic.
Exactly. It offered insights into complex processes like cell division, how genes are expressed in eukaryotes, how cellular components interact. It gives us a manageable window into the more intricate machinery of our own cells.
Okay, so from single cells, let's jump to the wonders of multicellular life. Now, before we get to the worms, it's worth mentioning the fruit fly, Drosophila melanogaster. That's a classic, right? It pioneered so much in invertebrate development.
Oh, absolutely. Drosophila the fruit fly was crucial, absolutely crucial for unraveling fundamental genetic principles like inheritance patterns.
Right, Mendel and all that.
And how genes determine basic body plan formation. It was key to understanding things like Homeotic genes, for example.
Homeotic genes?
They're like master control genes. They tell different body segments what they're supposed to become: head, thorax, abdomen, that sort of thing.
Oh, okay. Body plan blueprints.
Pretty much. So Drosophila truly set the stage for much of what we now know about animal development, even in more complex organisms.
And then we get to, well, a true superstar of transparency and simplicity in multicellular life. C. elegans, the worm.
Yes, the nematode worm.
It sounds incredibly useful, especially being so tiny, only about 1 millimeter long, with just about 1,000 somatic cells.
But what makes it so revolutionary for science?
C. elegans is just phenomenal. It was the first multicellular organism to have its complete genome sequenced.
Wow, the first.
The first, which tells you something about how accessible it is for genetic study.
Yeah.
But its key revolution really lies in its transparency.
It's see-through.
It's literally clear. So every cell can be observed under a microscope throughout development. You can watch every cell divide and move.
Exactly. Combine that with a life cycle of about 3 days from egg to adult and a lifespan of just 2-3 weeks. And you have this unparalleled system for studying development rapidly and comprehensively. You can literally watch an entire organism grow cell by cell.
So we can literally watch an entire organism grow, mapping every cell's destiny.
And every neural connection.
That's just an astonishing level of detail. How does understanding such a, well, simple animal translate to our own much more complex bodies?
Well, it means scientists have been able to achieve this amazing level of detail. They have a fully mapped cell lineage. Meaning they know the developmental fate of every single cell right from the fertilized egg.
Incredible.
And even more remarkably, they've mapped the complete nervous system, its connectome.
The wiring diagram?
Essentially, yes. The entire wiring diagram of its 302 neurons and all their synaptic connections. It's fully described.
Wow.
And this isn't just a static map. It's like a living blueprint. Knowing the precise connections lets scientists trace how specific behaviors arise from neural circuits. It offers foundational insights into basic neuroscience and developmental biology.
Insights we couldn't get elsewhere easily.
Exactly. Insights that are otherwise almost impossible to observe in more complex organisms. And crucially, many of its roughly 20,000 genes are homologous to human genes.
Meaning they're related evolutionarily.
Yes, they share a common evolutionary origin and often have similar functions. So the worm's simplicity plus that genetic connection is incredibly powerful.
Okay, let's level up again then to vertebrates, obviously closer to humans in evolutionary terms. We know that the frog and the zebrafish provide highly accessible vertebrate models. What unique advantages do these two offer?
Well, these two models offer distinct advantages for studying vertebrate development. They effectively bridge the gap between that invertebrate simplicity we just talked about and mammalian complexity. They let us study processes like how organs form, how complex nervous systems develop, all in a more human-like context, but still accessible.
Let's start with the frog, specifically Xenopus theanophlavis. It's a classical model, isn't it?
Yeah.
Used for a long time.
Yes, a cornerstone. Frogs are classical models for embryology for several really compelling reasons.
Such as?
Well, their large embryos and eggs aren't just easy to see, they're also easy to manipulate during early development.
You can physically work with them.
Yes, researchers can perform experimental manipulations like injecting substances directly into cells, doing microsurgery, even nuclear like cloning techniques without necessarily destroying the embryo.
Wow, that's robust.
It is. Plus, they have rapid development, which is invaluable for studying critical processes like cell division, how the body axis forms, head, tail, back, front, and the initial stages of organogenesis.
How organs start to form.
Exactly. How all our body parts begin to take shape.
That tolerance for manipulation sounds incredibly powerful. It makes you think about how much we could learn about subtle developmental defects that might lead to human conditions.
Exactly right. And that actually raises an important question, How do these very accessible features of frog development actually translate into understanding complex human development, especially those crucial early stages that are so difficult, ethically and practically, to observe directly in humans?
Yeah.
The fundamental pathways, though, are often conserved, shared between frogs and humans.
And then we have the zebrafish, Doniorario, another incredibly visual model. I think I've seen videos of them developing. Yeah. It's absolutely captivating.
Yes, the zebrafish is exceptional, largely because of its transparent embryos.
Like the worm.
Like the worm, but now in a vertebrate. This unique feature allows internal structures to be observed in real time under a microscope as they develop.
Wow.
And like the frog, it has rapid development. Major organs like the heart and brain form within just 24 to 48 hours.
That's incredibly fast.
It is. You can literally watch a heart start beating or a brain take shape before your eyes.
Plus, it's a prolific breeder, isn't it? The sources mentioned high fecundity.
Yes, very high. It produces hundreds of embryos at once.
Hundreds. That sounds like a massive advantage for certain types of research, right?
Precisely. That high fecundity makes it ideal for large-scale genetic and drug screens.
Testing lots of things at once.
Exactly. Researchers can test hundreds, maybe even thousands, of chemical compounds or genetic mutations simultaneously. It's a powerful tool for discovering new medicines or understanding the genetic basis of diseases.
And genetically accessible, too.
Yes. Its genetic accessibility makes it relatively easy to generate mutants and transgenic lines. So researchers can study specific gene functions or even model human disease states directly in the fish.
And crucially,
it's relevant to vertebrate biology. It shares many genes and developmental pathways with humans, which makes discoveries potentially directly applicable to our own biology.
Okay, finally, we arrive at the predominant mammalian model organism, the mouse. This is where it really hits home for understanding human health, I suppose, since we're both mammals.
Absolutely. This is the closest step in many ways.
How similar are we, truly?
Well, the mouse is indispensable because of its profound genetic similarity to humans. They share approximately 98% of their genes with us.
98%. Wow.
It's a huge overlap. This means many fundamental biological processes from the immune system and nervous system to metabolism and reproduction are conserved. They're largely the same.
So they're like our closest genetic cousins in the lab.
In essence, yes, which makes them an invaluable bridge for understanding human biology and disease.
So this incredible genetic similarity makes them highly relevant for modeling human diseases.
Extremely relevant.
And it's not just general relevance. The sources say similar mutations produce the same effect in human and mouse. That's a direct one-to-one connection sometimes.
Often, yes. And what's truly fascinating here, and incredibly powerful, is the sheer sophistication of the genetic manipulation tools available for mice.
More advanced than for other models.
Generally, yes, especially for mammals. We're talking about technologies like knockout mice, where scientists can precisely knock out a specific gene to see what happens.
Turn off a gene.
Right, or knock-in mice, where they introduce knock-in maybe a human gene or a specific human disease-causing mutation into the mouse genome.
Wow.
Then you have transgenic mice carrying foreign DNA, and of course, the incredibly precise CRISPR-Cas9 gene editing.
So you can make very specific changes.
Exactly. These tools allow scientists to precisely mimic human genetic conditions, creating models for diseases that directly mirror what we see in patients. This level of precision is, well, pretty much unmatched in other vertebrates used commonly in the lab.
And for practical lab work, they're pretty efficient too, right? Short life cycle and high reproductive rate.
Definitely helps speed things up. A gestation of only about 19-21 days leads to large litters, 6 to 12 pups, and the generation time is short. Lab mice typically live 1.5 to 3 years.
So you can study multiple generations relatively quickly.
Exactly. It accelerates research timelines considerably compared to, say, studying similar processes in larger mammals.
And beyond just the genetics.
Beyond genetics, their well-characterized biology is a huge asset. Their anatomy, physiology, developmental biology, it's all thoroughly documented. Decades of research.
We know a lot about them.
We know a ton about them, and this vast body of knowledge has led to a vast array of disease models for conditions like cancer, diabetes, Alzheimer's, cardiovascular disease, infectious diseases, just countless others.
They're the workhorses.
They really are the workhorses of preclinical research, where new therapies are often tested before they ever get near human trials.
And it's not just the individual mouse, is it? There's a whole infrastructure. The sources mention an entire community and resources supporting mouse research.
That's a really important point. Centralized resources like the Jackson Laboratory, for instance, provide thousands of specific mouse strains bred for particular research purposes.
Tailor-made mice.
Pretty much, along with extensive genomic databases and standardized protocols.
So everyone's on the same page.
Largely, yes. This robust infrastructure means mousework is widely reproducible and highly collaborative. Results can be compared across labs worldwide with more confidence.
Makes sense.
It's really no wonder there's even a bronze monument to the laboratory mouse. It's a testament to their immense, often unseen contribution to human health and, well, our understanding of life itself.
Wow, what a journey we've taken. From the simple, single-celled E. coli that laid the groundwork for molecular biology,
The very beginning.
through the transparent wonders of the C. elegans worm and the zebrafish that illuminated development,
Watching life unfold.
all the way to the genetically similar mouse. These model organisms are truly the unsung heroes behind so much of our biological knowledge. It's a constant reminder, isn't it, that big answers often come from looking at the small.
Indeed. Every medical breakthrough, really, every deeper understanding of human physiology or disease, it often stands on the shoulders of discoveries made first in these seemingly unassuming creatures.
Yeah.
They bridge that gap between the abstract biological questions we have and getting concrete answers. They let us test hypotheses in a living system before we apply that knowledge to humans.
It's efficient.
It's an elegant, efficient way to start deciphering the sheer complexity of life.
So what does this all mean for you listening in?
Yeah.
Well, the next time you hear about a new treatment for Alzheimer's or maybe a deeper understanding of human development or some disease, remember the critical role these models played. They're kind of a shortcut to being well-informed about the very foundation of life. And just a reminder that even the smallest creatures can unlock the biggest secrets.
And this raises, I think, an important question as we look forward. As technology keeps advancing, as our understanding of biology deepens, what new model organisms might emerge?
Ooh, interesting thought.
Or perhaps what novel ways might we use the existing ones, maybe combining them with AI or advanced imaging techniques to tackle the really grand biological challenges that still lie ahead?
Like what?
Well, think about areas like regenerative medicine or personalized genomics or even understanding the fundamental processes of aging. There's still so much to learn.
Something profound to ponder as you go about your day. Thanks for joining us on this deep dive into the incredible world of model organisms.